Okay, great. Thank you very much. So we'll have two lectures today. The first one is kind of a long one. We can go in one bit. Or we can break in a half. So we'll see how it goes. If you get tired and you want you need a break, just wave, okay? And then we will. If I see many people waving, then I'll stop and take a break. And then the second talk, on the other hand, is I think it's shorter, and we can do it in one piece. So we are going to do 1 + 1 or 2 halves + 1 today. And then again, once again, I encourage you not to memorize every single detail of what we have done, because it's not really so important, but rather to use it maybe to familiarize yourselves how we answer the questions and how the knowledge changes, and how the things that were obvious, the things that are obvious now were not obvious 5 years ago, and vice versa. Things that were well established 5 years ago, right now they're more complicated, or maybe they are made more simple. So of course I'm not going to repeat a previous talk. And also one more question: am I speaking too fast? Okay. That's good, because I could speak slower, but then the lecture is not going to be 45 minutes, but 1 hour and a half. So I'll then speak kind of the normal way. So in yesterday's talk, I introduced you to the cell cycle. We discussed that there are different phases of the cell cycle. That the G1 phase is particularly important because this is the time when the cells make a commitment for cell division. We also talked about the different cyclins which drive those cell cycle progression. We discussed that those cyclins are mainly regulatory subunits that bind cyclin-dependent kinases, and we talked that the cyclins and cyclin-dependent kinases operating in the G1 phase of the cell cycle are particularly important because they are receiving external signals and because they are deranging several cancers. And also at the biochemical levels, we talked that the D-type cyclins together with CDK4 and CDK6 phosphorylate the retinoblastoma protein, that this releases the E2F transcription factors. The E2F transcription factors induce several genes required for the S-phase entry and progression, but one of them is cyclin E. And then we talked that cyclin E further phosphorylates the retinoblastoma protein, leading to more release of E2F transcription factors and more induction of cyclin E. We also talked that the D-type cyclins also activate cyclin E complex through a kinase-independent mechanism by stealing from cyclin E inhibitor, which in non-proliferating cells inhibits the activity of cyclin E CDK2 kinase activity, and now this inhibitor is redistributed to cyclin D CDK4 and this activates cyclin E kinase activity. So in one view, well, very simplistic view, but somehow true, is that cyclin D CDK4 actually controls the activity of cyclin E, and then cyclin E is the major kinase that actually drives cell cycle progression, and then the function of cyclin D CDK4 CDK6 is to activate this kinase. Or stated differently, when we started this line of investigation that I'm going to discuss with you today, the dogma was the field is that actually cyclin E CDK2 is the major kinase that drives entry of cells into the S phase, and the function of cyclin D CDK4 is just this kinase is controlled by extracellular environment. So extracellular environment induces these cyclins, and this leads to activation of cyclin E CDK2. Cyclin E CDK2 being the driver of the cell cycle, and the cyclin D is the way that links this key kinase to the extracellular environment. And as we discussed, whereas the function of cyclin D at least seem to be narrow to activate cyclin E, we talked that once activated, cyclin E CDK2 performs several different functions. It phosphorylates retinoblastoma protein, but importantly, it also phosphorylates proteins that enter the cells into the S phase. It governs centrosomal duplication. It governs also histone biosynthesis, several other important functions. And then lastly, we talked that these three should be here. We talked that they are different. As we talk isoforms, or I think it's better to say different family members, there are 3 D-type cyclins and 2 E-type cyclins. And lastly, we discussed that the D-type cyclins are induced by mitogens, followed by induction of thank you very much, Anya. Thank you. Oh wow, thank you. So I'll repeat for Anya just what you missed, which is because this is the introduction. What I was starting with the introduction is that the D-type cyclins activate cyclin E-CDK2 through several independent mechanisms. One is phosphorylation of retinoblastoma protein, release of E2F transcription factors that then turn on cyclin E. Cyclin E further phosphorylates retinoblastoma protein, releases more E2F transcription factors, and induces more cyclin E. And the second I also discussed yesterday, the D-type cyclins steal the inhibitor from cyclin E-CDK2 to cyclin D-CDK4 and to activate the cyclin E. And then cyclin E, once activated, performs several other functions beyond the retinoblastoma protein, like phosphorylation of proteins required for entry of the cells into the S phase. And therefore, the thinking in the field when we started this work was that D-type cyclins actually serve as a links between cell environment and this key kinase, cyclin E. Because cyclin E is so important that once you activate cyclin E, that you trigger entry into the S phase, that you cannot just have uncontrolled or kind of going up and down in kind of uncontrolled fashion. So in order, so D-type cyclins together with RB were thought as a kind of layer of control to control the activity of cyclin E. Cyclin D being the controller of cyclin E through RB and E2Fs and through the titration, and cyclin E is the major one, this kind of the driver of the cell cycle progression. And then there are two, so that's where I was, where we came back. So there are two E-type cyclins, E1 and E2. They are associated with primarily with cyclin-dependent kinase 2. They are rapidly induced prior to the S phase entry. And this, and actually people have shown that if you take the cells and inject antibodies against cyclin E, by neutralize the function, you block the entry into the S phase. So this indicated that cyclin E is absolutely required for entry into the S phase. We can imagine, or people argue, we can imagine how you can proliferate without D-type cyclins, because D-type cyclins are just the controllers of this major kinase. But there is no proliferation without cyclin E, and indeed, if you inject antibodies or antisense oligos, you completely block S phase entry. This gene is like all cyclins and CDKs that operate in G1 phase. This gene is amplified and overexpressed in several cancers. And I mentioned that in addition to retinoblastoma phosphorylation, the E-type cyclins perform many different functions, and then E-type cyclins are absolutely essential for cell cycle progression. If you take the embryo and you look at the expression pattern of E-type cyclins with in situ hybridization, so the positive signal is in red, you will see that cyclin E1 and cyclin E2, either at day 9.5 or 11.5, are expressed exactly the same way. And again, red signal is positive, blue signal is just a background, and this is bromodesoxyuridine incorporation, which marks proliferating cells. And as you can see, every cell that is positive for BRDO, meaning that it's proliferating, is also going to be positive for cyclin E, consistent with the notion that cyclin E kind of is a major driver of cell proliferation. That you can, for example, see here. So we decided to, and we is Yang Geng, an instructor, so this is a faculty position in the lab originally from China, and Yan generated mice lacking cyclin E1. And she did it just by removing the key exons from the gene, and she found that these mice are viable, develop normally, and they have absolutely no phenotype. This is actually an example when in one slide, in 15 seconds, I am summarizing 2 years of somebody's work. But that's kind of the fact. We did this mouse, it takes 2 years to characterize, and there is no phenotype. So then we know that there is also cyclin E2. So Jan generated the second strain of mice lacking cyclin E2. Actually, there is a nice story about this, because we found out that there is a competing group, Bruno Amati, from Italy, that is knocking out cyclin E2, and we are knocking out cyclin E1. And rather than cutting each other's throats, we decided to actually exchange reagents. So we sent them the embryonic stem cells for cyclin E1 knockout. He sent us the construct for cyclin E2 knockout, so they could do the full experiment, and we could do the full experiment. So with the gene construct from them, we knocked out cyclin E2. We made mice again without cyclin E2 by deleting the key exons, and this time they're also viable, and some males are sterile, and they have a testicular hypoplasia and reduced sperm counts. So we talked talked yesterday about kind of requirement for cyclines, and of course the spermatogenesis is very important, because without this, you know, the animals would not propagate. But on the other hand, in the grand scheme of things, overwhelming majority of the cells can proliferate without E-type, without cyclin E2. So then we made the, I think, a reasonable logical hypothesis is that those two E-type cyclins perform overlapping redundant functions, and I showed you that almost every cell that has cyclin E1 has cyclin E2, so it makes sense. So we said, well, let's knock out both of them. Let's combine and make a double knockout. And when we did this, it turns out that cyclin E1, E2 knockout mice are embryonic lethal. So they are dying in embryo development, and this is actually a table how we do it. So we sacrifice the pregnant mother at day 10.25. So the pregnancy is gestation is 19 days. So at 10.25 we observe 7 double knockout mice, and 7 are expected. So it's exactly as you would expect from Mendelian ratio. And 10.5 we start seeing in brackets these are the embryos that are dead. So of course in controls we don't see dead embryos, but the knockout we start seeing dead embryos. Most of them are dead by 11.5, and by 12.5 every single embryo is dead. So now I'll show you the best looking embryo, the best looking meaning like, you know, the most, not the best looking like the most pretty, but the one that advanced the most far. And that's the one. So this is control, and this is double knockout embryo at day 10.75. So we could, Jan could have concluded at this point and said, okay, so this nicely shows that cyclin E1, they need to perform redundant functions, because if you knock out cyclin E1, you have no phenotype, you have knock out cyclin E2, you have almost no phenotype. And if you knock out both cyclins, then this is embryonic lethal. However, Jan looked very carefully, and what she found is that when she sectioned those embryos head to toe, she found absolutely no abnormalities. So those embryos are dying, and yet there's nothing abnormal with them. Could you suggest, do you have any ideas how it would be possible? How you can have a dying embryo if nothing here is wrong? Of course, one possibility is that we were not careful enough to find, but actually the truth is different. Do you have any ideas?
Maybe cyclin E is necessary for the next stages of development. Well,
but we are keeping them until they die. So this is the embryo that is going to die like very soon. So they are not dying because they know that they would die like a day or 2 days later. They are dying because there's something missing in them.
Problems with near tissues, not with embryo tissues, with motor-ish tissues.
Very good. Alexandra, right? Okay. Very good. So exactly. So this the problem is not with the embryo. The problem is with the extraembryonal tissues, like for example with placenta, which in part is embryonal derived. And this is actually the sections with the placenta, and with the arrow, we are marking here the so-called trophoblast giant cells. They are very, very big. You can see like this, how this is the nuclei of surrounding cells, very small ones, and these are the giant. And these are the trophoblast giant cells in knockout mice that are much smaller. And the reason why those nuclei are so big, it's not that they just grow in size, but they undergo the process called endoreplication, which is different. It's a different cell cycle that we just discussed. We just discussed in the cell cycle, the cells duplicate the genetic material, so it's S phase, and then they divide, M phase, S phase, M phase, S phase. So you always keep the same amount of DNA, as we call it 2N. But in the placenta, there is in those giant cells, there is endoreplication, is a different cell cycle when cells goes to S, but there is no mitosis. There's S and no mitosis. So they go from 2N to 4N to 8N, 16N. So they become polyploid. They become very big nuclei, which contain from other cells it would be abnormal amount of DNA. And indeed, you can actually we can quantify the amount of DNA per nucleus in control cells. This is in red, and put them in bin. So a normal cell has 2N or 4N. 4N is just in the G2 before mitosis, right? because they're 2n, 4n, 2n, 4n. But here you can see that in normal trophoblast giant cells, the cells have 30n, even 30 to 100n, or over 100n. I'm sorry, actually I'm showing you wrong. The black ones are controls, I'm sorry. So the black one is control. This is 30, 100, 300, and 300 to 900 n. So this is kind of a greatly increased, of course, amount of DNA. But in red are double knockouts, and in double knockouts, overwhelming majority is actually less than 30. So this is clearly skewed to the left. So we concluded that these mice are dying, that there is normal embryo development, abnormal placental development, and there is a paucity of trophoblast giant cells, and there is endoreplication problem. So do you think that based on this result we can conclude that these embryos are dying because of the placental problem, and then the cycle E is not required for embryo? What do you think? If you were the reviewer of a paper and I said, you know, we have shown that the embryo looks normal and the placenta looks abnormal and therefore they are dying because of placenta, would you believe it? No. Why you wouldn't believe it? Yeah? You said you wouldn't believe it. Why you wouldn't believe it? It's not enough. Right. So that's exactly the case. First of all, there could have been changes in embryo that we missed. The fact that those trophoblast giant cells are not big, I think we showed it convincingly, but that this is killing the embryo, it's pure speculation, right? You could argue maybe the embryo can live with those few giant cells, or maybe, as you were suggesting, maybe the embryo would be smaller, right? But it would be not enough to kill the embryo. So in order, how would you test this? How can we make sure that this is placenta that is killing those embryos?
Another placenta from another organism without knockout.
Right. Right. So that's exactly what we did. You want to provide your knockout embryos with wild-type placenta. And you want to see what happens. And there are different ways of doing this. But the way we did it is that we took the advantage of the method called tetraploid complementation. And this is a very nice method, so I'll describe it for you. We made our heterozygote mice, and then we sacrificed the pregnant females when they are 3 days after mating. So at that time they have very tiny embryos called blastocysts. And of course, one of the 16 of those blastocysts is going to be E1, E2 double knockouts, right? Because that's you will get all the possible genotypes of the embryo, and then we collect all the blastocysts, but then eventually we want these guys. And then from these we can have our own. We can derive embryonic stem cells, which are from here, and then we have our own embryonic stem cells, embryonal stem cells. In this case, it's E1, E2 double knockouts. Now we do actual tetraploid complementation. And the tetraploid complementation we do as follows. We take wild-type male and wild-type female. We put them into the same cage, they mate. The females become pregnant. We sacrifice the female very early after copulation, when the embryo is composed of 2 cells. And now we take this, each cell is 2n, this diploid cell, and now we put electric current, and then we fuse these cells. So now we are back to 1-cell embryo. This embryo is 1 cell, but it is a tetraploid, because we fuse 2n cells. And then we take this embryo and keep it for 3 days in incubator, a special medium, and we get a blastocyst. And this blastocyst is tetraploid. When you looked at this, you would not tell the difference. It looks like normal. But this abnormal blastocyst is because it's tetraploid. And therefore, if you take this blastocyst and put it into pregnant or pseudo-pregnant female, it cannot develop into embryo because it's tetraploid. It can only develop into placenta. So this we are providing wild-type placenta or tetraploid placenta, but it's wild-type. And now we take our knockout cells and inject into this placenta. Into this blastocyst, and then we implant them into recipient mice for further development. Do you understand? So the embryos that are developing is chimeric. The blue cells, tetraploid cells, are going to make placenta. So we are providing them with wild-type placenta. and the red cells that are going to be embryos. So the embryo is double knockout, but the placenta is wild type. And this is what happens. These mice are born, and at birth we can get E1, E2 double knockout. So we can rescue this lethality, and rather than dying in day 11, they go to birth. They go to birth, so as you can see, the overwhelming majority of the cells can proliferate without E-type cyclins, but they are still dying. And the reason they are still dying is that they have a heart defect that is not so important, but they have the septal defect, and also the arteries which are connecting to heart is not corrected, connected correctly. So you were right. I think we were right that the placenta is killing the embryo, but if we provide them with wild-type placenta, we can rescue it all the way, like 99%, but not 100%, because there is still heart that doesn't develop normally. So when the mouse are born and when the normal circulation occurs, those neonates are going to die. And lastly, something I will not show, but I'll just describe the experiment that. I showed you yesterday an experiment where we can do a knockout, inducible knockout in adult mouse. So we did it with cyclin E, and if you knock out cyclin E after the mouse is born, then now the mouse is perfectly happy. So the bottom line is that the cyclin E is required, first of all, for the placenta. You can bypass it by this tetraploid complementation. If you bypass by complementation, then you have the second roadblock, which is the heart problem, the swell problem, but it kills the mice. If you delete cyclin E afterwards, after the heart developed, well, now you have a perfectly normal mouse. So I think there are two important take-home messages. First of all, is that contrary to what was in the textbooks, cyclin E is not needed for proliferation. It's needed for proliferation of those trophoblast giant cells and of the heart cells, but apart from these, all other cells can proliferate without E-type cyclins. And this is good news for cancer therapy, because given the fact that these proteins play a role in cancer formation, maybe we can target these proteins in cancer. That's something we are working on, and I have nothing to report. But the second kind of surprising thing is how come the proliferation can occur without E-type cyclins. And we didn't understand it for many years. And we speculated, and the answer actually came from the work of my student from Poland, Ilona Kalaszczyńska, who was working on cyclin A. So now we need to move from cyclin E to cyclin A, and then in the end, I hope we'll understand why you can have a normal cell proliferation without cyclin E. So independently to this work, Ilona Kalaszczyńska was working on cyclin A, and the cyclin A is acting later in the S phase. So cyclin A is required as a S phase kinase. This is the kinase that drives cells for the S phase, binding through cyclin-dependent kinase 2, and then in the G2 phase, it also controls the entry into the M phase, and this is with another kinase partner called cyclin-dependent kinase 2. And then again, this was considered as an essential kinase. People actually pointed out that unlike those G1 cyclins, which are like two families expressed in the G1 phase, this is the only kinase that is present in the S phase. So it was shown to regulate DNA synthesis, induce initiation of DNA replication, and plays role in centrosome duplication and entry into the mitosis. And there are two A-type cyclins called cyclin A1 and A2, like D1, D2, D3, or like E1 and E2, but there's actually something different about those cyclins. Cyclin A1 is actually expressed only in the testes, and this is the paper that is not our work. I borrowed it from somebody else's publication, and this is, I think, Western blotting, where you can see that cyclin A1 is only in a test. This is test specific, and people made actually the knockout of cyclin A1, and there's no phenotype in anywhere except for male infertility, which makes sense. This is test specific. Cyclin A2, on the other hand, is expressed everywhere, and this is this S phase kinase. This is cyclin that drives the S phase. And Ilona actually did in situ hybridization. This is staining of the embryo for, again, the red signal is positive. So this is the signal of the embryo. There is no cyclin A1. this is testis for control, which expresses high level of cyclin A1, and this is the embryo, which expresses high level of cyclin A2. So this actually, this is written from the review written by kind of a very prominent cell cycle person, who said that cyclin A2 is an essential component of all embryonic and somatic cell cycle in mammals. And then people also rationalize and say, well, maybe the G1 cyclins, we can knock it out, but they are essentially two types of cyclins that are essential components of the cell cycle engine, and this is cyclin A and B. And this, in part, this notion was kind of rationalized because this is only cyclin or CDK expressed in the S phase, in part because of the antibody experiments. If you inject antibodies against cyclin A, you block entry into S phase. And also, people have knocked out cyclin A2, and unlike all our knockouts that can survive, like with D-type cyclins, there's no phenotype. In E, I mentioned to you that they are dying in day 11, so this is kind of quite good mid-gestational embryo. But cyclin A, as soon you get the embryo, you get some maternal RNA. As soon as maternal stocks are depleted, the embryos are dead. The embryos are dead very early before implantation. So this was yet again another indication that cyclin A is required for proliferation. But what Ilona kind of said is that she said, well, okay, well, if you have the early developing embryo and this embryo is dying very, very, very early, well, you cannot just say that this is such essential cyclin. It means that the cyclin is essential at this very, very, very early embryo. But what about if I knock out cyclin A later? Would cyclin A be required at later stages? So she generated a conditional knockout mouse, and I think I introduced yesterday conditional knockout, but I'll just repeat that basically if this is cyclin A gene, we put the short sequences called LOXP sites around the critical exons. And this is not going to change the expression of cyclin A, and therefore these mice are going to be wild type. However, whenever we express the Cre recombinase, that Cre recombinase is going to detect those LOXP sites, is going to delete everything that is going to be between LOXP sites, and is going to create a cyclin A null allele. So first of all, what Ilona did is she generated cyclin A2 conditional knockout mice. They are normal because cyclin A is expressed, and then she isolated fibroblasts from the embryo. She put Cre recombinase into them. This is Western blotting, these are the control cells. This is cyclin A2 flux, and this is cyclin A2 flux, conditional knockout together with the Cre recombinase. The expression of cyclin A2 is completely gone. Well, to our surprise, we found that these cells proliferate quite normally. So here we are, this is the days, and here we're counting the number of cells. In blue, this is increase in cell number of control cells, and in green is increase in number of cells when we knock out cyclin A2. There is no difference. Well, then we decide to stress those cells a little bit more, not just let them grow, but now we are going to remove the medium, so they stop growing, and now we are going to stimulate them with the medium, so we force them to enter the cell cycle, and now we'll see how they go. I'm sorry. So this is how they go to the cell cycle. So we are looking now at the incorporation of thymidine, which shows the cells going into the S phase. So in blue cells, at about 14 hours, they start going into the S phase, and now by 21 hours, 24 hours, most of the cells are in the S phase. If we knock out cyclin A, they go back to the cell cycle exactly the same way. Well, it was very surprising, and this is actually the staining with the propidium iodide and bromose oxyuridine. This is the fraction of G1 cells, S, and G2M, and here are the percentages, and they are very similar. Well, we said this is maybe not so very surprising, because there is another cyclin A called cyclin A1, and I did show you that cyclin A1 is expressed in testes, but maybe when we knock out cyclin A2, then now cyclin A1 becomes derepressed and could compensate for loss. So fortunately, we didn't have to make cyclin A1 knockout mice, because our collaborator Deborah Volgenbuth from Columbia generated cyclin A1 knockout. I mentioned they have no, I mean, they have very interesting phenotype from her because she's studying spermatogenesis. The males are sterile, but apart from this, these are the viable mice. So we could cross them. We could obtain a double knockout, where the cells cyclin A1 is knocked out and A2 is conditional, and now we can make fibroblasts. We can put crude recombinase. We can completely delete. So now we are deleting cyclin A1 and cyclin A2. There are no A-type cyclins there. And then we can check how they proliferate. This I just showed you before, and this is double knockout. So in red are control cells, and in blue are, I'm sorry, in blue are control cells, and in red are cells where we knocked out all A-type cyclins. They proliferate very nicely. There's no difference. Now we are going to force them to enter the cell cycle, and then once again you can see that they go back to the cell cycle identical fashion. And then the cell cycle profile is similar, although there is, as you may see, there is less cells in G1 and there is more cells in S phase. And actually this is quantification here. In blue are control cells. They typically have about 60% in G1 and then 20% in S phase, and then 25 in G2M, whereas the single knockout and double knockout, they have less cells in G1 and more cells in S phase. And actually we measure the length of the cell cycle. These are the control cells. This is how much they go through G1. G1 is the longest phase, then they go through the S, and they go through the G2M. In knockout, they go faster through G1 because they need more time to go through the S phase. So the S phase is not 100% normal without A-type cyclins. The S phase is longer, right? It's about longer by about an hour. Also, the G2M is longer. I mentioned that A-type cyclins are required for G2M entry. But overall, this is a very minor change because these two cell cycle phases are longer, the G1 is shorter, and overall the generation time, as you can see, is only half an hour longer than from controls, and this explains why they proliferate, normally. So now we have one mystery. I told you how the cells can proliferate without E-type cyclins, and now we are adding another mystery: how the cells can proliferate without A cyclins, and this is Ilona's problem. However, what we put them two and two together, and we said, you know, both E-type cyclins and A-type cyclins activate the same kinase, CDK2. And even though this is called the G1 cyclin, and this is called the S phase cyclin, they are controlling a very similar, processes. These control the entry into the S phase, and this controls progression through the S phase. The issue was that there was plenty of data and literature saying that those complexes phosphorylate different proteins. So the substrates are different from cyclin E CDK2 versus for cyclin A CDK2. So first of all, what we did, we looked at the cell cycle profile of control and of cyclin A knockout cells. And as you can see, most of the cells are identical. The level of cyclin D1 is identical, and D2, etc., etc. But what we noticed that cyclin E is actually upregulated in cyclin A knockouts. So when we knock out cyclin A, that cyclin E goes up. And this is actually shown here. This is the level of cyclin A in controls, and this is in knockouts. So in knockouts there is no cyclin A, but we have a higher level of cyclin E. Now, as you can see, the cyclin E and cyclin A have different patterns of expression. Cyclin E is expressed early S phase, and the cyclin A is expressed later. So just having a higher level of cyclin E doesn't help us, doesn't explain it, because you need to have the kinase through the whole S phase. And therefore we decided to very carefully look how the induction of cyclin E and cyclin A takes place in our cells. So these are the control cells, and as you can see, as you go from into the S phase, from the G1 into the S phase, the cyclin E becomes upregulated here, and then by 27 hours it goes down. So this is slightly up and then down, whereas cyclin A goes up later, right here. And this is shown in this one. This is the level of cyclin A. It goes up here, and in yellow, this level of cyclin E, it goes up and goes down. So as the cyclin E goes down, cyclin A goes up. In knockouts, in cyclin A knockouts, of course there is no cyclin A, but now we found that cyclin E, it goes up and then it stays high. And this actually quantification is in red. So again, in yellow, it's cyclin E in control mice. Control cell goes up and goes down. In red are the knockouts, cyclin E goes up and then stays on. So it's not that the level of cyclin E is higher. Cyclin E becomes now more broadly expressed. It's expressed like cyclin E, but then together with cyclin A. However, there was this notion that cyclin E and cyclin A control different phosphorylation of different substrates. So let's check whether this is true. So first of all, when we knock out cyclin A in our knockout cells, we still see activity. So this is the kinase activity of CDK2 and CDK1. This is controlled, and this is a knockout. So clearly, if we knock out A-type cyclins, then those kinases are still active because they're activated by cyclin E. So here we have several proteins using phosphor-specific antibodies, and some of those phosphosites are specific for cyclin E CDK2, some for cyclin A CDK2. I did mention to you that the notion was that these blue circles and red circles phosphorylate different proteins. But it turns out this is not the case, because this is phosphorylation of those substrates here in wild-type cells. This is in cyclin A knockout cells, and this is in cyclin E knockout cells, and they are perfectly normally phosphorylated. So this indicates that although some of those proteins, like for example this, was claimed to be cyclin A specific site, but this is not. You can knock out cyclin A, and this site is phosphorylated presumably by cyclin E. And also here, for example, this site is supposed to be cyclin E specific, but it's not true. If you knock out cyclin E, this site is phosphorylated by cyclin A. So our conclusion here is that although cyclin E and cyclin A containing complexes were thought to phosphorylate different proteins and to perform different functions, in fact, these are two very redundant classes of proteins. If you take cyclin E, then cyclin A can still phosphorylate all those proteins, and you have a normal proliferation. If you take cyclin A, cyclin E can phosphorylate proteins. Are you convinced by what I said, and can we just take questions, or do you think that maybe I didn't provide you enough data? Can we conclude that based on this, that cyclin E and cyclin A, well, I just told you, this protein is supposed to be cyclin E specific, but when you knock out cyclin E, it's phosphorylated normally. This one is supposed to be cyclin A specific, but when we knock out cyclin A, it is phosphorylated correctly. I told you that both of cyclins can activate CDK1 and CDK2, and we have shown that if we knock out these ones, that CDK1 and CDK2 are still active. Can we just conclude that E and A performs different overlapping functions, and just move to the second lecture? So what do you think? Yes. Okay, so you are not convinced. You think there could be there could be another protein. Okay, that's a very good point. Is anybody convinced? Okay, so I'm not convincing today, but you're absolutely right. This is a pure speculation, or as we call it, hand waving. Of course, maybe this is true, but there could be another protein. These proteins could be there could be cyclin E activating cyclin A, but there could be cyclin X activating, or maybe phosphorylation of these proteins is not needed. This is just a pure. This only shows that the studies which were claiming that phosphorylation of this site is cyclin A specific, well, that's clearly incorrect. So because you knock out cyclin A and this site is phosphorylated. Study that shows that this is cyclin E specific, well, of course it's not, because we knock out cyclin E and this is phosphorylated. But to say that those two cyclins are required because they phosphorylate the same proteins, it's pure hand waving, because as gentleman pointed out, there could be another protein. So, but how can we test this? How can we test that our hypothesis is correct? Yeah, all four. You mean? Yeah, exactly. So knock out all four, And that's what we did. This is quadruple knockout. So, but this is a complicated experiment because I just mentioned to you, so now we are going to knock out E and A type cyclins. We are going to do quadruple knockout, and we are going to see what happens. But it's a difficult experiment because if I told you that once we knock out E type cyclins, we have, because that's what you want us to do, right? You have A1 knockout, A2 flux, which we are going to delete, E1 knockout, E2 knockout. But I just told you that just this E1, E2 knockout, it's embryonic lethal. So if we combine them at the best, we are going to have this embryonic lethality, probably earlier. But even if not, the best scenario is that we are going to get early embryos, and there is not much we can do with them. So how can we do this cross, but have something meaningful? Sorry?
Conditional knockout of E cycle.
Conditional knockout of E cycle is a great idea. We didn't have it at that time. Because of course you can have conditional knockout of E and you don't have to worry about this. We didn't have conditional knockout at that time. Well, I just told you that in order to go from here to here, we did the tetraploid complementation with cyclin E knockout embryonic stem cells. So we did tetraploid complementation, but this is tetraploid complementation with quadruple knockout ES cells. So Ilona mated these mice, E1, E2, A1 minus, A2 flux. Right? So that's what we have to do. We have to get these cells, quadruple knockout embryonic stem cells, and then inject them into traploid blastocysts. Okay, so this is what Ilona did. She mated A1 knockouts, A2 flux, E1 minus, E2 minus, and then she obtained the quadruple knockout embryonic stem cells. She then takes the quadruple knockout embryonic stem cells, but cyclin A2 is fluxed. So this way, so this is conditional expressed. We get quadruple knockout embryonic stem cells. We inject them into tetraploid blastocysts. We can get the embryos day 13.5. From these we get fibroblasts. The A1 is knocked out. A2 is conditional, so it's being expressed. E1 is knockout. E2 is knockout. Now we put recombinase. We get rid of cyclin A expression, and we see what happens. And now we see the proliferation stops. So this is the time in incorporation and controls, and this is why we put Cre. It doesn't go to 0, because when we put Cre, we cannot put it to 100% of cells. So this is once it goes down, but it's not extinguished. This is another way of measuring, and this is the growth of cells. These are the cells before, and this is the shutdown of cyclin A. And now this is the kinase activity. I told you that we knock out cyclin A, that there is still CDK1 and CDK2 kinase activity, and the gentleman in the green shirt said, well, said maybe there is another protein. So it's great thinking, but luckily there is not another protein, because you can see that once we extinguish all of them, all four, then now the kinase activity is gone. So in summary, it turns out that cyclin E and cyclin A on their own are dispensable for proliferation, because they perform very similar functions in phosphorylating proteins, and it's only if we ablate all E and A type cyclins that we arrest cell proliferation. How are we doing with time? How many minutes I've? But of course I'll finish when I finish. I have another 15 minutes about this subject. Do you want to continue, or do you want to take a 5 minutes break? Huh? Continue. Okay. Sorry? Is it okay if I continue?
Yes, I'm going to break before the second.
Yes. Okay, but can you stand 15 minutes more about this and the second talk? Yeah? Very good. Okay, so I'll skip this, and I'll just tell you that so far I have shown you that if we knock out cyclin A in fibroblasts, that there is no proliferation defect. And I told you that we have to knock out cyclin A and cyclin E to see anything. And I left you, or would have left you, with the notion that cyclin E and cyclin A is required. They kind of play redundant function. But now, since you decided to stay 15 minutes longer, then I'll tell you that this was not correct. I mean, it was correct in fibroblasts, right? But it doesn't mean that it's true on other cell types. And this, I think, is a good indication that that once you learn something in one cell type, you have to be very careful to generalize it into all other compartments. And if you were not so resilient, and if you didn't want to stay another 15 minutes, maybe you would have left this room and said, well, Peter Sicинski told us that cyclin E and cyclin A perform redundant functions. Cyclin A is not required for proliferation because cyclin E is there. But Peter Sicинski told you only about fibroblasts. And fibroblasts is just one cell type, not the most maybe interesting one, although that's the one that is mostly studied in the cell cycle field. So most of the models that people are making, including ourselves, are from the cell cycle field. But now we are going to have a reality check. And we are going to say, what happens if you knock out cyclin A in other compartments? Is it going to be the same story or different story? And this is done in bone marrow. And the way we do it is that we have cyclin A1 minus, the phenotype only in the smaller testes. The A2 is conditional, so it's being expressed. And the Cre recombinase is MX Cre. And this is hematopoietic specific Cre. So when we inject mice with a chemical called poly-I, poly-C, we activate Cre in hematopoietic cells, in particular in bone marrow, and we see what happens. And when we see what happens, the mice are dying. This is the control mice, and in red are cyclin A2 conditional knockout, and in green are cyclin A2 conditional together with cyclin A1 minus, because we want always to, we were expecting there is going to be no phenotype based on fibroblasts, and therefore Ilona said, okay, well, let's just combine it with cyclin A1, because of course there is not going to be a phenotype here. We already showed that in the fibroblasts cyclin A and E perform redundant functions, so it's kind of a control experiment for bone marrow. Let's show in a conclusive way that bone marrow is the same. But no, bone marrow is different, because bone marrow, something happens to the bone marrow, because these mice are dying, and indeed when you look at the cell numbers, you can see that this is red blood cells in control, and this is a knockout mice. So clearly they are very anemic. And when you look at the bone marrow, you can see the number of bone marrow cells in knockouts goes down dramatically, and this is the quantification. So what happens to these cells? Well, these cells stop proliferating. But what's interesting is that the cells are the cells that are so this is just a bone marrow showing that all population from bone marrow are gone. These are granulocytes, this is macrophages, these are erythroid cells. In the bone marrow, all the cells are generated from hematopoietic stem cells, which they generate common myeloid progenitors, granulocyte, macrophage, monocyte progenitors, and then more differentiated cells. What we found is that when you knock out cyclin A, that these cells, the hematopoietic stem cells, disappear. And this actually here, these are the numbers of hematopoietic stem cells. This is control, and this is knockouts. So they're gone. Like I think that the difference is like 1,000-fold, and these are the very immature progenitors. Whereas when you look at more mature cells, they are initially normal. So this is, for example, GR1 MAC positive cells. These are the controls, and this is cyclin A knockout, and this is double knockout. So for some reason, something happens to those stem cells, that those stem cells seem to require cyclin A. So to test it kind of in rigorous way, what we did with the help from Koichi Akashi, who had a stem cell lab in our institute, because now he left, we took hematopoietic stem cells, we sorted them, and then we put them one cell per well, so we can follow this cell. And then we put the Cre recombinase to delete cyclin A, and then we keep them, we give them the cytokines to boost their proliferation. And then do we see kind of a large clusters of cells. So first of all, we have a control experiment where cyclin A1 is plus minus and cyclin A2 is flox plus. So this heterozygote. When we put Cre recombinase, two different things can happen. Either Cre recombinase is not going to work, So we are going to get back A1 plus minus A2 flux plus, and we saw two colonies like this. Or the Cre combinase is going to work, is going to delete the flux allele, and it's going to be cycling A2 plus minus. This we get in most of the colonies. Now we are going to do the same with the real experiment. So we are going to have cycling A1 minus minus, because we want to knock out A cyclins, and cycling A2 is flux deleted. One allele is deleted, and the second is flux. Why is one is already deleted? Because we make it easier for Cre recombinase, right? Here for the control experiment, we are asking Cre recombinase to delete one allele, flux allele, right? So here we are helping this as well. One allele is already deleted, and we are only asking to delete the second allele. And what happened is that every single colony that we see comes from non-deleted cells. We can never get this doubly deleted colonies. So for some reason, stem cells don't like cycling A to be deleted. We can never see those colonies. This is hematopoietic stem cells. So let's look at another stem cell kind. And another stem cell kind is embryonic stem cells. This is the cells that we use to make mice from. I mentioned to you that we can make a mouse from a single embryonic stem cells, and when we grow them, this is actually the colony of these cells. So now we are going to put Cre and see what happens. So in the first experiment, we have control experiment A1 plus minus A2 flux plus. We put Cre, and either the Cre is not going to work and we get back the same genotype, or Cre is going to work and is going to be deleted. It works most of the time. Now we are going to take A1 minus minus A2 flux flux. We are going to put Cre. Either the Cre is not going to work and we get back this genotype, or we can delete one allele, and this happens here, or we delete both alleles. Never happens. So then we say, well, maybe Cre is not strong enough. So let's take these cells. So one allele is already deleted. We just need to delete one. We take these cells. They are A2 flux deleted. We put Cre, and either the Cre is not going to work, or Cre is going to work, and we can never get this. And therefore we concluded that proliferation of fibroblasts does not depend on cycling A, but proliferation of hematopoietic stem cells and embryonic cells critically depends on cycling A. I will stop here without going into molecular analysis. In particular, that our interpretation, when this work was published, we interpreted that the fibroblasts do not require cycling A for proliferation, because cycling E is compensating for the function. In stem cells, on the other hand, it turns out that that cycling A plays absolutely essential role, because if we knock out cycling A, we could never see those colonies of embryonic stem cells, or never see the colonies of hematopoietic stem. And we concluded that the proliferation of stem cells, for some reason, depends on cycling A. We actually rationalized why it is, and we are hypothesizing that in fibroblasts there is high level of cycling A and high level of cycling E. So it makes sense that if you remove cycling A, cycling E can compensate, and vice versa. But in several stem cell compartment, cycling A is high and cycling E is low. So we were speculating that this probably makes it dependent on cycling A. But this is not correct, because now we found that there's a paper that it came out in Nature, I think 2 weeks ago, from Weni Wei lab, although our names are on this because we actually collaborated with them, and they have found that in several compartments, cycling A plays a role in survival of cells, not in proliferation, but it plays in pro-survival, but phosphorylating the AKT kinase. And actually, together with them, working with them, or told by them, we have shown, and this is in the paper, that actually, if you knock out cycling A in stem cells, because here this experiment shows you that you never get any colonies. Well, we interpreted that we never get any colonies because they don't proliferate, right? So you cannot see single cells. But now we did a situation when we have stem cells, and then we can turn off cycling A. And what happens, these cells are dying. So the thinking that cycling A is required for proliferation was not correct. It's required for survival of stem cells. And this is why I was preparing to talk for you. I corrected and I wrote here the survival. Okay, so this is my acknowledgement slide, and the person who did it is Ilona. Where's Ilona? Well, I'm sorry, but she's not in this picture. However, so now I remember what happened. She went back to Poland for a few months, and actually there was Ilona, another Ilona Kondratiuk, another student who was helping with this project. But Ilona's picture is not here, I'm sorry. And Deborah Volgemuth from Columbia gave us the mice, and then the lab of Koichi Akashi helped us with the stem cells analysis. Okay, so I stop here and maybe take the questions to this talk, then we have a break, and then we're going to a second lecture.